Our session over the next hour is called Understanding Human Health at the Individual Level. And I'm excited to be able to welcome some real leaders in the field from Silicon Valley to be able to talk to this subject. I'm going to kick things off by just talking for a few moments on a couple of themes, and then I'm going to pass over to my panelists because we're really here to hear from them and then have the opportunity as well to ask them questions. All right, so this is me. And these are my disclosures. You'll see I have relationships with a number of different companies. We have collaborations as well as advisory roles. I wanted to share with you, though, I think a much more telling conflict of interest statement that came from Zach Kohani from Twitter, of all places. He said, non-financial conflict is the dark matter of academic bias. Bias from pride in your public ideas can be much greater than monetarily induced bias. I completely endorse that position. And I've never found a very short 140-character way of stating that. So I thought I'd just use his quote, hashtag self-criticism, hashtag glass houses. So there we go. That's the disclosures. This is a blast from the past now. Our former President Obama kind of launched the idea of precision medicine, as I mentioned this morning in my introductory remarks, with his usage of the term at the State of the Union address and the launching of the Precision Medicine Initiative after that. And there are many representatives, of course, from that great initiative as it moves forward at the meeting here. And he said, I want the country that eliminated polio and mapped the human genome to lead a new era of medicine, one that delivers the right treatment at the right time. And this is pretty much how we define it. We want to predict, prevent, and then treat much more precisely. And so we think about precision health as well as precision medicine. But for precision medicine, we also have to think about accuracy. And as many of you know, these two words are semantically distinct. We also need precision medicine to be accurate. We need both precision and accuracy. And I wanted to spend just a few minutes as an introduction to the session talking about what I mean by that. And there's a lot of information on this slide, but I wanted to speak to just one or 2 points in it just for a few moments. Because as we currently think of the application of next-generation sequencing to medicine, there are a number of areas of great success. I mentioned in my remarks this morning are now pretty confidently found 25 to 35% solve rate for undiagnosed disease and for rare and Mendelian disease. We talked about the application that you'll hear more about shortly from the panel in oncology and the excitement of immuno-oncology that is all around us. We also heard about pharmacogenomics. But for all those applications, it's important that we pay attention to accuracy because most of these technologies that we're using and the algorithms that have been used to interpret the data were developed in an era of discovery. And the downside with an algorithm, if it doesn't work quite right in a discovery context, is that you don't make the discovery, or rather you don't make the next discovery. You probably made 10 and published the paper and that's fine. But when we move that into a medicine context, then this is why accuracy becomes much more important, because if your algorithm isn't optimal, if your technology isn't optimal, then now we're dealing with people's lives, individuals' lives, large extended families' lives. And so that explains a lot of the focus of my group at Stanford on the idea of accuracy within next-generation sequencing. And this is a summary from a paper that we published last year talking about some of the challenges. And predominantly, of course, our technology is based around short reads, and many of the challenges in short reads for diagnostics are laid out here. If we're talking about the Illumina technology, for example, there are the challenges of high GC content, the enrichment by capture, which we all use in Exome products, is great, but of course the capture is based on the reference sequence. And we're interested not so much in the reference sequence, but in where the sequence of the DNA we're looking at changes and is different from the reference sequence. So we're already setting ourselves up for a challenge. We have to use a reference sequence still because of the computational challenge of de novo assembly, and we often end up with a large percentage of unmapped reads. We miss variants also because the reference sequence is from a group of people who were real people and had real variants. We have challenges because many of our diseases are caused by repeat tracks. And if you have a read that's only 100 base pairs long and you have a repeat track that's causing disease at 400 or 500 base pairs, it's very challenging, not impossible, but very challenging to work out how long that repeat track really is. Paralogous sequence is something that keeps me up at night. 5% of the genome likely is able to originate redundant sequence. So even if you have perfect technology and perfect sequencing, you're still going to be uncertain where to place a very large number of reads in the genome. And that's before we even get to the highly polymorphic regions such as HLA, the olfactory region, and others. Our algorithms are great for single nucleotide variation. They're not really nearly so good for insertions and deletions, small ones, or larger ones in the form of structural variation. Major challenge. And then when we think about the data we have to think, in general, we talk about where the variant call file that represents the patient is different from the reference. As I mentioned, the reference has variants in it. And if you want to make calls for precision medicine, you're going to need to make calls everywhere, and we get back into a larger data problem at that time. Finally, what do the variants actually mean? There's challenges there too, and we're only just beginning to put together as a community the resources for data sharing that we just heard about in the last session to allow maximization of the ability to connect people across continents, to be able to connect variants that we found in different patients with good treatments and good inference of causality. Well, I wanted to give you one example of one approach to some of those challenges, because it's not right to stand up and explain there are a lot of challenges and not offer something. And so I just wanted to tell you just for a few minutes before I hand over about this patient of mine. And he is a patient that I've known for many years. His history is laid out on the left. He's had 3 open heart surgeries. And this is his heart that you see beating with an echocardiogram. And you can see, I don't have a laser pointer, but you can see there's a little ball-shaped thing wobbling around in the middle. And I can't really point to it, but I think you'll see that's basically a tumor in his heart. And the little small one that you see at the bottom left of this picture here is not actually his major problem, but rather the kind of large ball that comes in and out at the bottom right of the upper figure, shown in the 3D below. And these are cardiac myxomata. These are tumors of the heart. And he seemed to have a condition that had a striking resemblance to something called Carney syndrome, which had many of the features that are laid out here. But the clinical testing for that, which was done in the earlier days with Sanger sequencing, was negative for the one gene that was known. We had run a whole genome with short read sequencing, in fact, on him as well to try to find the answer. And of course, as always, we find needles among stacks of needles, but we weren't able to find the answer from the short read sequencing. So we thought that it would be worthwhile trying a new approach, in this case using the Pacific Biosciences technology to look at long read sequencing. And this is what was done. This was just done in the last few months and just really shown in public for the first time a month or two ago. This was actually one prior to the current chemistry, so this could actually be done on fewer smart cells now for this amount of data. You'll see the read length N50 is 9,614 base pairs from this run that was done in October. The average variant size is shown here. You can see the LU repeats, and you can see again the very large size of the variants that were called. 6,000 to 7,000 deletions and insertions greater than 50 base pairs. So there's a large dark matter here that we haven't seen yet that we're going to have to get our arms around if we want to make sense of these kinds of insertions and deletions. Oh, I'm going to go back one. In this case, as you can see, our filtering was pretty straightforward. Again, we don't have an exact or a nomad for these kinds of large structural variations. There are databases out there. We used the NA12878 healthy control, which was a useful filter. Then we had to look to see which of the insertions and deletions overlapped a gene known to cause disease in OMIM. And we were happy to see that when we looked at that, one of the genes that we found was the PRKR1A gene, which is the gene known to be associated with Carney syndrome. You can see it here. The average coverage was 10-fold across the genome, but in this region was only 4-fold, which is small, but you can see that exactly 2 reads show the exact deletion that is shown here, and exactly 2 reads do not. And you can see it in more detail at the bottom left of the screen. So with a very small number of reads, you can actually, with very high sensitivity and specificity, pick up large structural variation. This wouldn't be good, of course, for calling single nucleotide variants across the genome. This is very specifically aimed at filling in the black hole that was provided by the earlier sequencing. But the question, of course, is now you know where it is, can you go back and look at the short reads and find it? And this is what's shown in panel C here. And of course, with the benefit of knowing where to look now, we can see there is actually a signal there. And within the very long list of possible structural variants provided by the algorithms that we ran on the short read sequencing, indeed, this was there. But the problem, as always, is picking the right needle from among the stack of other needles that are there. So we felt that this was one option and one approach, the long read sequencing, towards an answer to some of those challenges that I mentioned in the first slide. This work is on bioRxiv. A huge shout out for bioRxiv and preprints. They're making a hit now after spending their heyday in math and physics. They're now hitting biology. And we are putting all our data from now on, all our papers, onto bioRxiv before they are submitted to a peer-reviewed journal. So that's all I wanted to say. Really, I'm here just to introduce this amazing panel. And then if we have time for some questions in between the talks, we'll certainly do that. So I'm going to pass over now. And it's my great pleasure to introduce a friend and colleague, Rich Chen, who's the Chief Scientific Officer of Personalis. Rich. Great to see you.
Thanks, Ewan. So I want to thank the organizers and Ewan for inviting me to speak to you today about the work that we and our partners are doing to develop this new area of personalized cancer vaccines. And so immunotherapy has been transformational in a number of areas in cancer, including one of great interest to me, melanoma. Jimmy Carter, as you know, was diagnosed with malignant melanoma, metastatic melanoma, in 2015 and received one of these immunotherapies and subsequently went into remission. And so it's really the dramatic individual results like this have led to this explosion in immunotherapy and immuno-oncology. In 2015, there were about 150 clinical trials in checkpoint blockade. That number quadrupled in 2016, in addition to a huge number of new trials in other areas of immunotherapy, including vaccines and also now new personalized vaccines, which I'll talk about today. But one of the key issues is that for most cancers, really only a fraction of the patients respond to immunotherapy, including checkpoint blockers. And so there's intense interest in understanding, are there additional methods or approaches that can increase the response rates in these patients, either as monotherapy or as adjunct to things like checkpoint blockade. One observation is that any one-size-fits-all approach, including checkpoint blockade, but also any targeted therapy approaches, are inherently limited in cancer because every tumor is different biologically and genetically, and it's changing dynamically, and it's changing in very significant ways. So one of those ways that tumors are different is that they're unique in their foreignness to the immune system. So each tumor has a set of mutations unique to that tumor. Those mutations code for or lead to tumor-specific antigens, which we call neoantigens, that can elicit an immune response. So you have the more mutations you have, the more neoantigens you have, the more foreign the tumor looks to the body, and the more likely you're going to have an immune response to the tumor. And so this is a study done in 2015 that showed that patients with high neoantigen burden responded better to immunotherapy than those with low neoantigen burden. The other uniqueness about tumors is that they each evolve different mechanisms to escape the immune system. So one example of this is that tumors can downregulate the expression of HLA or MHC that present the very neoantigens that we were just talking about. So if those HLA or MHC are downregulated, the neoantigens don't get presented to the immune system and no immune response is triggered. Tumor microenvironments can also vary very dramatically between each tumor. The mix of cytotoxic, helper, Tregs, myeloid cells, and the activating and inhibitory cytokines that they all secrete can dramatically change how someone's going to respond to an immunotherapy. So the question is this: how and can we harness the unique biology of each tumor to create a truly personalized therapeutic? And this is indeed what we and our partners are working on, is developing these neoantigen-based personalized cancer vaccines where we actually comprehensively characterize the biology of the tumor, the neoantigens, and then we use that information to actually, in real time, rationally design a vaccine specific to that tumor and that tumor's neoantigens. And also, actually including information about how that tumor is evolving in terms of escape mechanisms and also tumor microenvironment. So using all that information together to create a personalized approach. Now, it turns out that neoantigens are actually a very, in theory, a very attractive vaccine target. They're less attenuated by host central tolerance. They're less likely to trigger an autoimmune response. You have the benefit of being able to target multiple neoantigens per tumor, which is helpful for avoiding tumor escape. And it's really only recently that we've begun to have the technologies that are necessary to be able to predict these neoantigens. So how does this work? So it starts with biopsying the patient's tumor. Then we use our ACE immunoID platform to comprehensively sequence and characterize the genetics of the tumor. That data then flows into the neoantigen prediction and tumor escape algorithms. And then our vaccine partners then synthesize a specific vaccine that targets those neoantigens. That gets administered with checkpoint blockade or adjuvants, and then the patient receives that personalized therapeutic. And the key here is this actually happens for each and every patient uniquely. The technology underlying the ACE immunoID platform, as well as some of the very practical considerations around the regulatory and validation that you need to make this practical at the bedside. So at the heart of the ACE immunoID platform is our patented ACE technology to achieve clinical grade sequencing. This gets to the accuracy issues that Ewan was talking about. That allows us to comprehensively and deeply sequence with our ACE exome and transcriptome that generates the DNA and RNA data that we can then use in our informatics and neoantigen algorithms to begin to identify the neoantigens and also begin to identify the mechanisms of tumor escape that they then can actually be used rationally to design these personalized vaccines. So the process starts with the sample prep, where we address some of the very thorny issues with difficult sample types that are involved in cancer. It can be very difficult to extract nucleic acids from formalin-fixed paraffin-embedded tissue. Then we use our ACE exome and transcriptome to really deeply sequence these tumors. That leads into our informatics algorithms, where we predict things like the epitope prediction. We do Hla typing. We actually then can predict the binding of those epitopes to the MHC complex. That leads to a neoantigen list. And then we overlay information about how similar to self-antigens those neoantigens are, the immunogenicity of those neoantigens, and then whether those neoantigens are actually being expressed, because if it's not expressed, it's not going to be immunogenic. So we can go from tens of thousands of mutations to a handful of neoepitopes. One of the key issues with these neoantigen, predicting these antigens, is the sensitivity and specificity of the neoantigen prediction. So studies have actually shown that with prior methods, neoantigen prediction can be a little bit of a dicey proposition. Not all neoantigens actually validate when you actually test to see if they're immunogenic. And that's a problem. If you're creating a vaccine that targets 10 of these neoantigens and 50% of them don't actually pan out, then you've wasted half your vaccine. So with some of the key efforts at Personalis are around improving these predictions by starting with the sequencing, then improving the machine learning and informatics around this, then actually doing very robust validation. So starting with the sequencing, one of the misconceptions that people have is that standard sequencing covers all genes completely, and that's absolutely not true. There are systematic biases in the sequences, regions of high GC that are traditionally very difficult to sequence, and it can leave gaps in major exons for important genes, and that can lead to missed neoantigens. So we've developed technology called ACE that actually fills in these gaps, and you can see the ACE coverage is much more uniform and gives you more complete coverage over each and every gene in the exome. And the green and the blue together is what we were able to cover with our ACE approach. And we've shown that this is actually very critical for clinical applications, much less therapeutic applications like this. ACE performs at a higher finishing rate than a lot of the other exome approaches out there, and also even whole genome in certain circumstances. And we published the results of this in Genome Medicine. But the key question here is how does this impact neoantigens? So this is an example. This is a gene that can harbor neoantigens, SIN1. And what we've done is actually predicted the neoantigens that can actually occur all along this gene. And you can see that there are some neoantigen deserts, so there are little gaps where you don't really get neoantigens. But for the most part, these neoantigens can occur almost anywhere along this gene. So if you actually overlay the gene coverage from a standard sequencing approach in blue here, you can see in the boxed region that there are a whole host of neoantigens that would be missed because of poor coverage. And with an ACE approach, we can actually fill in those gaps and make sure we don't actually miss those. The other really important thing with immuno-oncology is that the RNA-seq data is absolutely important. You want to know if these neoantigens are getting expressed, and ACE also improves that performance as well. Okay, so the question is then, that's sequencing, and it has to be, it's absolutely critical for making this work. But how do we identify and then select these neoantigens? And the trick here is going from tens of thousands of potential peptide candidates for neoantigens and getting down and understanding which of those actually bind to the MHC class 1 and class 2 complexes, because if it doesn't bind to the complexes, it's not going to trigger an immune response. So we and others have built neural networks to predict this binding. We can take potential peptides, and then that becomes input into our neural networks. And then the output of the neural network is how strongly is this peptide likely to bind to the MHC class 1 and class 2 complexes. Now, with any machine learning approach, it's very sensitive to the quality and quantity of the training data that's available. And that's one of the challenges. So here I've shown the publicly available data for training for about 100 of these HLA alleles. And you can see on the right-hand side, there's some alleles that have a fair amount of training data, but actually there's a vast majority actually don't. So to address this, we're actually building our own peptidomics data set through mass spec for training these neural networks. That's going to greatly expand not just the positive training set, but also the negative training set for these HLA across a broader swath of HLA alleles. The other thing we're doing is actually expanding beyond just the MHC1 and 2 binding prediction. You can see here that upstream of the binding, there's a whole gauntlet of the antigen presenting machinery that the peptide has to be able to make its way through to actually be presented. And so incorporating data about the antigen presenting machinery into our data is going to be very helpful for improving the predictions. So with any process like this that you're going to apply in a therapeutic setting, validation is absolutely key. One of the things that we put a lot of effort into is analytically validating our sequencing exome and transcriptome by taking standard FFPE samples and also cell lines, mixing them in different dilution series to get at our limits of detection, because tumors are heterogeneous. They come in 10% tumor, 20% tumor, 30% tumor, or less in some point. So you have to know what your limits of detection are. And what we've shown is actually best-in-class high sensitivity for SNPs, indels, and fusions, which are the key pieces of what can turn into these neoantigens at minor allele frequencies of 10% or above. You can see here we've done this for both the exome and also calling variants on the transcriptome. But the other piece of validation that's also really critical is the neoantigen prediction itself. And so we've kind of approached this problem in two parts. We've done in silico validation by assembling 23 previously established immunogenic neoantigens across different tumor types. And we spike that into our immunoID data set and look to see if our algorithms could then pull those back out among the sea of all other possibilities. And indeed, we were able to pull out 22 of those 23 known neoantigens, which suggests a fairly reasonable sensitivity of our approach. But sensitivity is really only a piece of the puzzle. Obviously, there's a lot more to do here. Specificity is also quite important. One thing we're doing is actually establishing more gold standard data sets, working with our collaborators to take our neoantigen predictions and actually validate whether they trigger a T-cell response in the actual laboratory. And then we're also working with the Tesla Consortium, which is part of the Parker Institute, working with 20 other academic groups and 10 industry groups to create these gold standard data sets so we can better validate these critical predictions. One other component of this that's absolutely critical to making this all work is that you have to have accurate HLA typing from the exome. And we've validated hundreds of HLA alleles now across MHC class 1 and 2. correlated that with the clinical HLA testing, and shown that we have actually very high concordance. So now I've talked about the sequencing and the new antigens, but the other piece of the biology that's critical here is understanding tumor escape mechanisms. This is a paper by Blank et al. In Science of 2016. They coined the term this immunogram, which I think is a very nice way to think about it. And essentially, we're building our ACE immunoID platform to elucidate the immunogram more comprehensively across things like the neoantigen burden, but also what are the immune infiltrates that are in the tumor itself? What is the HLA type? Is it getting downregulated? The cytolytic activity, JAK1, JAK2, TAP expression, the list goes on. And the notion here is that this information can come together to really rank and better understand which new antigens are likely to be the ones that pan out for a personalized vaccine. So what I haven't touched much on is what our partners are doing to then build the vaccines that actually deliver the new antigens, which is a whole set of other talks themselves. But quickly, there are multiple approaches that are being tried. Folks are using DNA-based approaches, messenger RNA, synthetic long peptides, recombinant viral approaches, with or without adjuvants, with or without checkpoint blockade, really striving to achieve not just a CD8 response here, but also a memory response with CD4, and also obviously looking for ability to get tumor regression. And the exciting thing here is that the clinical trials that we're partnering with are just beginning in this, so this is a very exciting time. The last 2 or 3 slides, I'm just going to talk about some of the very practical issues and taking a complex process like this to the bedside. And so you can see here, going from an FFPE block to the neoantigen predictions is a very complex process. We want to be able to do this reproducibly, traceably at every step within 30 to 60 days. Personalis has built a CLIA and CAP laboratory to process 10,000 samples, panels, exomes, whole genomes. We have a Illumina X5 system. We have the sample prep, robotics, and laboratory LIMS to manage this process. And we built an enterprise clinical NGS system to enable us to precisely control this process from start to finish, to lock down the components, to have traceability for compliance, and allow us to scale to massive volumes. And so the final slide talks about the regulatory considerations with the FDA for this really uncharted new area of therapeutics. And I've talked about some of the things we're doing, but we're also close to implementing an ISO 15189 on the way to QSR, which is required for precise design control for these processes. And we're initiating now a filing with the FDA that will support the use of ACE immunoID with personalized vaccines. So my last slide is quickly in summary. This is really still early days, but we think this truly personalized approach can be really synergistic with existing immunotherapeutic approaches. The biology and understanding that at an individual tumor level is absolutely critical for making this work with new antigens and tumor escape. We're working on improving, validating, and really industrializing the assays and the processes and the informatics required to power these vaccines. Thank you very much.
Great, thank you, Rich, so much. That was wonderful. I think given time, we're going to move on with the panel members. If we have time, we'll have some questions at the end, and if not, we'll have an opportunity to talk to the panel just immediately afterwards. So one of the joys of having genomics as part of precision medicine and healthcare is that the technology companies that are all around us here have got interested because it's big data. And I think we are as close as we could be to Google headquarters. And so we're very happy to welcome now Jessica Megger, who's the Chief Medical Officer at Verily. Thanks.
Thank you. It is an absolute pleasure to be here. And I want us to really think about our title today. And what we've been asked to reflect on is how do we understand both health and disease at the individual level? And what I want to start with is the concept that as a physician, I think that we have always been trying to personalize care. I think anyone who has sat across from a patient, either in a clinic or at their bedside, we integrate where's that person coming from, both from a social perspective, from a medical perspective, and also from all of the data that we receive. But we've reached this very interesting point where the convergence of technology and healthcare is now accelerating. And you've heard from even the talks this morning how things have really hit an inflection point. And it was with that in mind that I personally turned from working, and I'll tell you a little bit about my journey, from working in clinical trials and in pharmacogenetics to now being embedded in a technology company. And when I first joined, I joined Google X, which was right down the road. I saw in the lobby here that there was, I believe, one of the driverless cars. And people sometimes ask, what do cars have in common with healthcare? And I think it's that these are areas that have tons of information. And if we do certain things, we can actually make a difference for people's lives. So with that in mind, the story I'll tell today is to think a bit about how do we actually collect various forms of information, what are the tools that we have to organize it, and importantly, how do we bring it back to patients? And I think the last panel so nicely articulated that in order to have individual insights, we have to put patients truly at the center of this. And that's where I'll conclude. So information in the healthcare space is everywhere. We have medical records. I certainly, when I learned to practice, if you can see in the far left-hand corner, this is the type of data exchange we were using. We have now medications that are in certain electronic health records. We have large imaging files, devices, everything that we heard about today from the genetics profile. And what got me particularly interested in personalized medicine and understanding and organizing some of this information was taking care of patients with heart attacks. And I was part of a research group where we were looking at the next generation of therapies and how do we actually make a difference for patients once they've had this illness. And one of the most commonly prescribed medications for these patients is clopidogrel. Clopidogrel is also known as Plavix. It's a prodrug that has to undergo different biologic transformation to lead to an active component. And what was interesting is we started to realize that not everyone was responding similarly to this medication, which I just told you is one of the most commonly prescribed medications worldwide. And we and others got quite interested in understanding were there certain genetic variants that would end up coding for different CYP enzymes differently. And ultimately, what we ended up finding is if you subset people based on their genetics, and the group in red I'm showing you here is people who carried a particular genetic variant in one of their CYP enzymes, other genes that encode the enzymes, and the blue line shows you non-carriers. And what we observed is among patients treated with clopidogrel after a heart attack, there was about a 50% increased risk of cardiovascular death and minor stroke among people with particular genetic variants. And for me, this was incredibly eye-opening. We ended up forging many partnerships with people in the room today, looking to see, does this replicate? And I think Ewan, in his introductory comments for us today, said that we are at a point where we're starting to understand, is there a role for genetics broadly? I will say it's interesting the number of different pieces that we now need to translate to actually bring all of these interesting points into clinical care is one that we're still going to have to discuss. And you heard even in the talks during this panel that point genetics is a way to go, but this idea of really taking whole genome sequencing is certainly the wave of the future. Now, I could spend the talk continuing to focus on that because I do think large data analytics is going to be incredibly relevant there. But I've been asked to also focus on personalized medicine broadly. And again, thinking the title is we're trying to get down to the individual. And so we know that information is not only coming from someone's genotype, but there's also the downstream analytes, so something like glucose. And there's even what we're starting to call not only genotype and phenotype, but what is the behavior type? And so it's to plant the seed that this information that we're going to be personalizing is going to be coming from a host of sources. What I'm showing you here is an example of a pretty standard glucometer. My grandfather was a diabetic, and for those of you who know me, I like to tell stories. And so when he originally was diagnosed with diabetes, he would take out a composition notebook and write all of his values down. And then he would go to his primary care doctor and bring this very diligent notebook. And it became clear to me pretty quickly that that really wasn't going to be an effective way of transmitting that information. Nowadays, we're at a point where it's actually relatively easy to connect glucometers to transmitted devices and actually share it with providers. But that's just the tip of the iceberg. There is a whole next generation of new connected devices. And what I'm showing you here on the left-hand side of the slide is a next generation prototype of incredibly small continuous glucose monitors. And on the right-hand side is showing you a prototype of a glucose sensing contact lens. So this idea of could we actually instrument pieces of equipment that we use every single day? And the reason that we're going to see more and more of these devices is because there has been a huge push in getting small, biocompatible electronics. And what I'm showing you on the bottom of the slide is just how small these electronics are getting, whether it's an integrated circuit, BLE, or connectivity devices. But this is going to be a change in the way that we collect information, going from punctuated episodic information to continuous information. And I think it's very important for the people in the room today to start to think about where do we think that information is going to be medically relevant? Because just because we can capture something continuously, we certainly have to ask, should we be doing that? But we are going to have more and more opportunity as we move forward. So that was this idea. We talked about genomics. We've talked about maybe measuring something like an analyte like glucose. But there's also going to be continuous digital signals that we're going to have to understand how do we integrate. And instead of basing a diagnostic recommendation or changing someone's therapy based on the few minutes that I, as a cardiologist, might spend with my patient in the four walls of the hospital, how do we integrate everything that's happening to a person every day of their life? So I'm showing you an example here of a spoon. This is a Liftware spoon. It's a tremor stabilizing spoon. It's used to help people actually eat. It works, it's 70% cancellation of tremors, works using different axes. But one piece I think that we can't ignore is it also provides continuous data for someone who's eating 3 times a day. And we have to ask ourselves, would it be interesting to understand if we were titrating medications, for example, for a tremor, would we want to integrate that data as we think about titrating medications, again, beyond just those punctuated episodes when we see someone in clinic? And these are going to be the types of tools that we'll be able to work with moving forward. So the second concept, if we are getting all of this data, it starts to put us in a different category. We're no longer going to be thinking about even large files, but if they're episodic, we don't need to think about how do you actually stream real-time raw data. And there certainly is a large revolution going now trying to tap into a lot of the architecture and infrastructure that has been built for other big data industries. And we certainly could talk about that in the question and answer. But there has been a transformation. And if you think about 2010, this idea of how did you compute, a lot of people would really focus on where did you compute? Where were you actually putting certain files? The data was on premise. It was relatively hard to access. I think for those of you who are here for the last panel, Harlan Krumholtz nicely articulated that the data may be out there, but it's hard for people to get access. I think that was an important point. But we're now moving to a different era where there's collective intelligence and distributed computing. Data can be stored in the cloud. There can be sharing as long as we get the right access controls. And even when I turn to colleagues who are on the panel today, we have a great partnership with Stanford, really trying to think about this next generation of collaboration and understanding data. So it's no longer where we compute, but it's how we compute. And that's going to start to change the tools that we use to understand big data and actually personalize care. So I'm going to give you a tangible example to make this something that's concrete, because this particular panel is talking about an individual patient. So we know there's a deep learning revolution. What does that actually mean? It means that we're using layered network architecture to try to extract different features. Much of this technology has been really championed around image recognition. So for those of you who go online, or for those of you who are YouTube enthusiasts, you know there are a great deal of cat videos that are out there. Thank goodness for cats, because it's pushed the technology to a degree that we can now use it for healthcare. So how do we use image recognition for healthcare? How do we translate all of those insights? Well, I'm showing you a different panel of diabetic retinopathy. And the images on the left show a patient without any diabetic retinopathy, and the images on the left-hand side are showing you patients with severe diabetic retinopathy. Diabetic retinopathy is one of the leading causes of blindness in patients with diabetes. In many cases, we're asking ophthalmologists or optometrists to go and read these different images and provide results. But we know that ophthalmologists and optometrists have a limited amount of time, particularly in developing countries. So the idea is, how can we use that same technology around image recognition and apply it to medical case conditions? And this is a paper that we worked on and thinking about deep learning algorithms to try to detect digital retinopathy. And the take-home message was deep learning algorithms had relatively high sensitivity and high specificity for detecting diabetic retinopathy. And this is just a proof of concept, but it's another way of using and organizing data to make it actionable for the individual. If we can start to use these techniques, and I think in the introductory session this morning, we heard about using particular cases to try to say, how can we push these new technologies in order to be much more democratic with care? And this, I think, is one example where we have these new technologies that we can start to use. So the final piece, as we think about this journey and think about the story of the individual, is not only collecting information, not only using next generation tools to organize it, but how do we get individuals to be engaged? And this isn't something where it really is meant to be flippant about starting with people. I mean this very genuinely. Unless we get people engaged and interested in being at the center of this, then we will not learn more about health and disease because there won't be that impetus. Times are changing, data is changing. We heard a call in the last session to really empower people with their data. The other idea is designing with the individual in mind. We always have to start with that individual and think, if there is data that we're providing, are we going to make it actionable and useful? The examples in the last panel, thinking about ways to change different diagnoses around cancer and tailoring therapies, to me, put that individual right at the center. Another area that we've been thinking about is how do you actually get data, something like the continuous glucose monitor, into the hands of patients? And I want to pause and say, has anyone in the room heard of Night Scout? Is this something, raise of hands, any Night Scout followers? Okay, I see a handful of people. This is a really interesting story about how information is getting into the hands of individuals and how it actually may be patients and parents who are going to be driving this revelation. So Night Scout ended up coming from this concept of an individual who had a 4-year-old son, and his 4-year-old son was diagnosed with type 1 diabetes. And he was able to get a continuous glucose monitor, which gave them the information they needed when they were at home. The challenge happened that when the 4-year-old was away from them, the parents could no longer get the real-time information. Now, as it turns out, this parent happened to be quite savvy and was a programmer and was able to go in and reroute the data and make it available to himself. So essentially hacked and created his own solution. He then posted this solution on Twitter, and it caught on. And other people and other parents said, this is incredibly important. Can you show me how to do this? And I think this is a real call to action for us to realize that when we make it important, and when it is important to an individual, then that is where we are truly going to change the parameters around understanding health and disease at that level. And to me, this makes us think about care not only in the 4 walls of a hospital or a clinic, but how do we partner with individuals to make a difference? So I think this is just, as I mentioned, the tip of the iceberg. But if we end up understanding how to collect relevant information, how to use tools to organize it, and how to put the individuals in the center, we're going to be doing our best effort to really individualize insights. So I thank you for your attention and look forward to questions at the end.
Thank you so much, Jessica. That's great. We're going to move on now to the healthcare delivery aspect. I'll do the handing over of the. I knew I was going to have to introduce Somi, and I talked to him a few weeks ago about how to pronounce his name, and he even gave me a recording of it, which I couldn't find last night. He mentioned to me that even his kids can't pronounce his name. Anyway, Somi is the director of bioinformatics at the Clinical Genome Service at Stanford Healthcare. Somi Utiramarua.
Yeah.
How did I do?
Good.
Okay.
Utiramirur. So that'll be a challenge at the end of for pronunciation. So first of all, I'd like to start by thanking Ewan for the opportunity to present some of the challenges that we have faced and overcome, and the lessons learned as we went about establishing our bootstrapping a precision medicine initiative at Stanford. Definitely in this audience, if I ask for a definition of precision medicine, probably I'll get as many different answers as there are people in this room. But in very essence, what we try to do is try to capture as much of the patient data as possible, whether it be molecular sequencing data, data from other diagnostics such as wearables, aggregated with population level data, whether it be different patient cohorts, healthy individuals like Exact or Nomad, and analyze it all together to arrive at the precise molecular mechanism of the disease in the patient. And finally, and hopefully, tailor-make the treatment not only based on the molecular etiology of the disease, but genetic makeup of the patient itself. So at Stanford, the Clinical Genomic Service, which is a partnership between the Stanford School of Medicine and both the Children's and the Adults Hospital system, we try to use exome and genome level sequencing, and we are focused primarily on inherited diseases, rare childhood diseases, inherited cancer, familial heart diseases, and neurological disorders. So when you are using genome or even exome level sequencing to answer rare diseases, the challenges you face are extremely different from the questions or challenges that you face when you are asking precise questions as to the presence or absence of variants in well-characterized genes, as in the case of gene panels. The principle among which is, are we ever going to be reimbursed for genome or exome level sequencing? But we all recently heard that everybody is going to be covered and covered beautifully. So I think that challenge is a solved problem. So the second most important challenge is that we see that the diagnostic yield that across various disease thing is roughly 30%. So how do we improve upon that? So it behooves us to use different technologies like long-read sequencing that you had mentioned earlier, linked reads and other genetic analysis methods in order to be able to improve upon that. And also we will touch upon the importance of data sharing and use of multiple annotation sources during variant classification stage. So before we go about incorporating some of these novel methods or sequencing technologies into the clinical diagnostics, the principal challenge that we face is that we have to meet the fundamental requirement that the assay be repeatable, reproducible, and traceable, which is not a big challenge if your assay looks something like this. Is one instrument which is tightly coupled with your electronic medical record system. Samples go in and your results go back to your EMR. This is the direction that all NGS platforms are moving towards, but unfortunately, as of today, we are far from being there except for single gene panels or tests like such as CFTR. So if you look at exome or genome level sequencing assays, this is how it looks like. We have a number of laboratory instruments that we use in our assay. So that requires efficient sample tracking through all the laboratory methods. We use a number of analytical methods in order to be able to call and find those variants. Believe me, if anybody tells there is a single pipeline or single method that can help answer all your questions, there isn't one. And as I mentioned, there are a number of annotation sources that are spread across different institutions throughout the world, which we absolutely have to incorporate during the decision-making process in order to be able to achieve the proper diagnostic yield. Here, I'm not even mentioning the hardware layer, which brings about whole new complexity when you try to have reproducible results. So until NGS assays becomes as routine and as reliable as a blood test from a drop of blood, we need some mechanisms in order to be able to reproduce our results and have it traceability. So here we introduce what we call as Genome Information Management System, which is essentially extension of the lab workflow management system, and most importantly, a bioinformatics workflow manager, which completely abstracts away our dependency on the hardware layer, allowing us to reproduce our assays regardless of whether we run on a distributed cluster system or any one of our cloud platforms. So this is essence of our Genome Information Management System, which runs on Google Cloud Platform, which tightly couples with the EMR system, and that is the only system that our users, our physicians interface with, that interfaces with all the annotation sources, whether it be public or proprietary annotation sources, in order to drive our decision, and also has lab workflow management for sample tracking. So the key component of our Genome Information Management System is Loom, which is essentially a bioinformatics workflow manager, which allows us to have reproducible analysis and assay results. So this is critically important when you have genome-level sequencing assays where you have hundreds of steps that are distributed across several systems, different algorithms and different methods that the results have to be reproduced. So what Loom tries to achieve is to be able to separate the analysis from the execution of those methods. So in essence, what Loom tries to do is when you give an analysis pipeline, it deploys a virtual machine on the Google Cloud Platform. This example is running on GCP, deploys the storage container, which has all the software components that is necessary for executing a single step, and manages the process and provides complete file provenance in order to be able to assure traceability and auditability of the results, which is so critical for a clinical diagnostic assay. So next, I'll go over some of a couple of the pilot case examples. Yuan already was one of them. It highlights the need for use of novel sequencing methods and analytical methods into clinical diagnostic assays, and also the importance of data sharing. The first case is the one that Yuan mentioned. Here, I want to highlight the fact that people consider that alignment and variant calling are solved problems, and they definitely are not. Primarily because, as in the case example that Yuan mentioned, this is a 2 kb deletion of whole exon in PKR18 gene that we have some signal in short-read sequencing, and current methods are able to pick up this variant, but the specificity of these methods are far from being able to be used in a clinical diagnostic setting. So there is a lot of work that's still yet to be done in order to improve upon these methods, either for short-read sequencing or being able to develop novel methods for incorporating long-read sequencing technologies into a clinical diagnostic assay. The next is a very important and useful case that highlights the importance of data sharing in a clinical diagnostics. This is a case of 2 siblings in a family where they're both affected by cerebral ataxia, and they were sequenced using whole genome sequencing in our lab, and we found a homozygous loss of function variant in SNX14 gene in both affected children. But at the point when this case was analyzed in our lab, the SNX14 gene was not implicated in cerebral ataxia, and it was through pure serendipity that our medical directors had conversation with other physicians who had also seen in their patient cohorts loss of function variant in SNX14 gene, which led to leaked classification of this variant from BUS to pathogenic, solving a diagnostic odyssey for this family. So that should not be the way, the modus operandi for having to diagnose and curate variants. So if you were to do this diagnosis today through our system, we would be able to report upon all the existing variants that have been published for SNX14 gene that is likely pathogenic or causal for cerebral ataxia. So the importance here that I want to highlight is that there are data sharing efforts that leads us to help identify these variants, and also the relevant literature that leads to behind this evidence. I assure you that not all evidences or not all literature are equal, and we need to carefully consider the evidence that we use in variant classification. And finally, I want to highlight this aspect of our system, which is also developed with other consortiums, is to be able to arrive at an alternative diagnosis and all from pure phenotypes, and also to be able to see whether a particular phenotype is segregating for a particular disease diagnosis or not. Finally, I'd like to thank both our medical directors, Yuan and Jason, for the vision, and the management behind Stanford School of Medicine and both the adult and the children's hospital for this project. Thank you.
Well, thank you so much, Soumya. And I think we do have just a few minutes here at the end to take some questions before lunch, or maybe one question that we could have each of you answer. So I've decided, other than having everyone in the country have beautiful coverage, if I was to give you a magic wand and you wanted to move us from where we are today to the point that the title of the session suggests, that we can truly do medicine at the individual level, what would you do? If you could change one thing, tell me what it would be.
Okay, I can go first. Well, I think I would try to, if I could wave the magic wand, I think aligning the incentives for this type of individualized care among the providers, among the the insurance companies, among the healthcare systems, and also the individuals, I think that's a problem, and the pharmaceutical companies. So yeah. I think the incentives are not always all aligned. I think that's a fundamental issue. but maybe waving the magic wand and getting some alignment there, I think, is a critical problem we have to solve. Because, I mean, individualized care, that doesn't always mean better, bigger market size.
Right.
But then there you have the cost of society if you don't individualize care, and there's all these, vaccines, you now have checkpoint blockers, you have all these opportunities to treat patients. You have to get smart about which ones get those treatments, otherwise it's going to be unaffordable. So finding ways to align those incentives, I think, is really important.
Makes sense. And reimbursement. Jessica?
I think we have to figure out how to show value to the individual. There's so many things that if we think about ourselves in our daily life, if we feel that there's value, we engage. And I think it's pretty easy to look at technologies that we're already using because we need to use the maps because as it turns out, my navigational prowess has gone downhill since maps has been introduced. But how do we show that same type of value when it comes to healthcare? And again, there's some really tangible examples, and the last panel highlighted those. How do we extend beyond that point where it's only of value when you're quite ill? How do we actually show value upstream? Because I think a lot of the talks today are around preventing illness, but unless we show that value, we will not make, I think, a lot of headway at the individual level.
Yeah, no, that's great. Just getting some investment early. And then, Somi, last word to you.
So, primarily what I feel is this is a large gap in the data from genome sequencing to panel level sequencing. So there's a large amount of investment in sequencing healthy individuals and cohorts from NIH and from other research institutions. But when it comes to clinical data, there's a large gaps in patient cohorts that we're not able to learn from by simply doing gene panel or exome level sequencing at this point. So having extra investment into sequencing certain patient cohorts would be valuable information so that we can show the benefit before we get reimbursements for being able to make it into a routine clinical care.
Yeah. Excellent comments. Well, I'm afraid we have come to the end. Thank you for joining us today, and please join me in thanking the panel for joining us today.